Hello friends, today we are going to discuss about one of the important topic in system physiology, about physiology of digestion. About physiology of digestion. You know, food is a fundamental and basic requirement for all the animals for energy and to repair and for growth of the cells. The thing is, food consists of varied substances. For instance, minerals, the water, the vitamins that can be absorbed directly into the cells. But complex biomacromolecules like carbohydrates, proteins, and fats that cannot be absorbed in the present form has to convert into simple diffusible or absorbable forms, what we call digestion. That means digestion is nothing but the process of conversion of complex non-diffusible food substances into the simple diffusible and or absorbable forms. Therefore, here the objective of digestive system is to fulfill this task. And digestive system consists of a basic digestive tract or alimentary canal and associated digestive glands. Here the most important fundamental thing is the digestion will not take place in one go. It will take place in a sequential fashion. Say for example, the proteins will digest it at one place, say stomach, and the rest in other place. What we call sequential digestion or progressive digestion. Here digestive tract, it's about basic, starts from mouth, what we call anterior opening, then the oral cavity or buccal cavity, pharynx, esophagus, then stomach. The stomach again, cardiac, fundic and pyloric stomach. Then that leads to small intestine. That means duodenum, jejunum, ileum. Then large intestine, cecum, colon, rectum, and ultimately the posterior opening called anus. This is about digestive tract. Associating the digestive tract, there are digestive glands which are essential for digestion. Say for example, salivary glands. Say the gastric glands, the pancreas, the liver. The thing is, say the gastric glands and the intestinal glands, what we call Brunner's glands and crypts of Leberkuhn. That means the secretions of Brunner's glands and crypts of Leberkuhn leads to succus entericus, what we call intestinal juice. These two present, that means gastric glands and intestinal glands present in the wall of the alimentary canal, whereas salivary glands, pancreas and liver, they are extra alimentary canal glands. That means they are present, that means they associate with the digestive tract. It's about the basic fundamentals of digestive tract. So the objective of digestion is to convert complex non-diffusible food substances into that simple absorbable or diffusible food substances. Okay. Objective of digestion is conversion of complex non-diffusible food substances into their simple diffusible or absorbable forms. It's the objective of this is the definition of digestion. And in the today's tutorial, we'll be discussing about how the digestion will take place in different parts of the digestive tract or alimentary canal, and how different glands, that means the enzymatic process, will involve in the same. Actually this digestion is of two kinds. The mechanical digestion, two, the chemical digestion. Mechanical digestion is, say for example, chewing. That means mastication, and peristalsis. That is the churning movement, say churning movements. Chemical digestion is by enzymes of different digestive glands. It's an overall picture of physiology of digestion. That means it takes place in two ways: one mechanical digestion and two chemical digestion. Mechanical digestion, chewing. We knew that the teeth will help in chewing the food, but we're going to mastication. Then the peristalsis. That means here the alternate contraction and relaxation of the smooth vessels of the wall of the alimentary canal, starting from the esophagus up to rectum. Usually the peristalsis are associated with the esophagus and up to rectum. And how this plays, and this also plays an important role. That means the churning movements, particularly in the stomach, where the churning movements are so powerful that it facilitates the digestion. That means mechanical digestion. Whereas chemical, simply by enzymes, or discuss now. Enzymes are the hydrolytic enzymes which will break down the biomolecules, the complex macromolecules into its simple form. Say for example, carbohydrate, say a starch, actually is a polymer. It has to break down into disaccharides, say maltose, the sucrose, the lactose. Say maltose again has to convert it into simple monosaccharides. Say the one maltose leads to two glucose molecules. Then the sucrose, glucose plus fructose. Then the lactose, one glucose plus galactose. That means associated with respect to enzymes will be done to complete this process. It's about carbohydrates, okay? And different enzymes will be responsible to break down sequentially. I told you now, it's about sequential digestion. Digestion will not take place in one go, okay? It's about, different steps. Then we'll be discussing about digestion, how digestion takes place in different parts. The thing is, there are three major hubs, what we call hotspots of digestion, major places in the alimentary canal. Say digestion in buccal cavity, then digestion in stomach, then small intestine. Digestion in buccal cavity or oral cavity, then the stomach, then the small intestine. These are the three major places where the major part of the digestion will take place. Okay. The first, the digestion in buccal cavity or oral cavity. The mouth is the anterior opening of the alimentary canal. The food we eat will goes inside the buccal cavity or oral cavity, and will there it will be subjected to mastication. That means the teeth will be cutting the form. That means cutting down the food. That means chewing of food. Teeth, tongue, along with saliva help in mixing of food. Here the mucus of the saliva help in lubrication and adhering the masticated food particles to form a bolus. What we call, the product that we get in buccal cavity is bolus. Bolus means the lubricated, the adhered part of food substances that has been subjected to mastication under the impact of mucus of saliva is bolus. Okay, here saliva is very important. No, I told you already. Mechanical digestion and chemical digestion. At some place, both will take place. Here, how we'll discuss. Okay, first the mastication is over by teeth. That means teeth, tongue, and saliva. That means salivary glands will release saliva. Okay, and these contains different components like electrolytes. Then most important thing, salivary amylase. This is very important. Salivary amylase, lysozyme. So electrolyte like so, Na plus, K plus, like that. Okay. So here the thing is, the salivary glands will release saliva. It consists of electrolytes. Then, and most important enzyme, salivary amylase, what we call ptyalin. Then lysozyme. These are the three essential components of saliva, and apart from this, the mucus help in creation of bolus. I told you already. So that what is the objective of this digestion in buccal cavity? To create bolus. That means mechanical digestion by means of churning, that means chewing activity by teeth, and also making it into bolus, then facilitating to deglutition or swallowing. That means the second important activity, deglutition or swallowing. Just imagine, we chew the food with the help of teeth. The tongue, that means the saliva will be released, will create in the form of a bolus, and it also release salivary amylase, lysozyme enzymes, and after that the bolus will be subjected to deglutition, that means swallowing. That means it will carry to pharynx, then from pharynx it goes to esophagus by deglutition. We swallow the food so that it can reach the esophagus. From pharynx to esophagus by deglutition. This is very important. Okay. Degradation, that means the buccal cavity to pharynx and pharynx to esophagus. Okay, where degradation is followed. So these are the two objectives. Then the mechanical digestion, to the degradation. Okay, the two objectives of buccal cavity and how the mechanical digestion takes place. You know, the teeth will chew the food, the tongue will mix the food, that means the help of saliva. Here the saliva contain different enzymes like saliva amylase, that enzyme, and the most important pylene. We have to discuss this part. You know, here carbohydrates will break down at the rate of 30%. No proteins and fats. We have already discussed now. Sequential digestion, that means some particular substances may take place in X or Y part and some other in Z or A part. It depends. Okay. Say for example, how the issue ends. Say the carbohydrates. That means say the starch. Starch is a polymer. It will subjected to ptyalin. That means salivary amylase. Okay. The pH is 6.8 to form maltose. Maltose is a disaccharide. Okay, disaccharide. The first is at the rate of 30%. 30, that means 30% of carbohydrate digestion. That means the conversion of starch, which is a polymer, into disaccharides like maltose at the rate of 30% will take place in buccal cavity under the catalytic activity of ptyalin or salivary amylase. This is very important. We got it, maltose. Only just carbohydrates. Okay. But as lysozyme is an antibacterial enzyme. It will kill unwanted bacteria. Okay. This is very important. No, starch, ptyalin, the under-substituent ptyalin form maltose, disaccharide. These are the then after that degradation, that means swallowing into esophagus. Then the peristalsis. The next movement is digestion in stomach. The next part. I told you not degradation or swallowing. How it will take place. The next zone, digestion in stomach. I've already discussed now. The wall of the alimentary canal starting from esophagus to rectum will be subjected to peristalsis. Peristalsis here, the thing is, you know, is like alternate contraction and relaxation of the smooth muscles, so that the food will propel down, that will propel down the tract, say from esophagus to stomach. In stomach, the peristalsis are so powerful that it facilitates mechanical digestion. That means the smooth muscles are, say for example, contract, relax, so that the food substances, say like this, just imagine, this is a normal tract, say this contracts and relaxes the food, so that it will go down from esophagus to stomach. From esophagus to stomach. This is very important. Then ultimately the food goes to stomach. Then what happens in stomach? We have already discussed. Stomach is a major place of digestion for proteins. That means different biomolecules and different biomolecules at different parts. And first what happens when it goes down to stomach? After the peristaltic subjection to stomach, here the food will be mixed with gastric juice. That means secreted by gastric glands. That means acidic gastric juice. Here the pH is 1.8. Most important thing is the pH is 1.8. Why 1.8 pH is required? Because to break down, that means here there is an enzyme called pepsin. Pepsin will break down the most powerful bond, what we call peptide bonds. To break down the peptide bond, that means for the catalytic activity of pepsin, this highly concentrated pH of 1.8 is essential. It will break down the peptide bond. That means to break down the peptide bond, concentrated environment medium is required. Done by pH 1.8 at the rate of pH 1.8, and it will maintained by the HCl, hydrochloric acid. This HCl, that means present one of the gastric juice contains one of the components is HCl. This HCl maintains the acidic medium and the pepsin act and break down. This is the basic fundamental requirement. At the same time, you know, the food which goes down, and that means we will store in stomach for 4 to 5 hours, will mix up with acetic gastric juice. By churning movement, the muscular wall, I told you already, the stomach wall is so powerful that it facilitates the mechanical digestion to form chyme, what we call chyme. The food mixed with the acidic gastric juice subjected to the churning movements of stomach wall. Okay, is chyme. Okay, then this chyme will be subjected to different activities. I told you already, different enzymes plays an important role, and we will discuss about that one by one. The most important enzymes with respect to gastric juice are, say the pepsinogen, the prorenin. But the thing is, this pepsinogen and prorenin are inactive enzymes. They have to convert into active forms. How they convert into active forms? The pepsinogen here under the activity of HCl converts into active form called pepsin. The same in case of prorenin also, HCl convert into renin. The most important thing you may ask, since the HCl pH is very powerful, does it not affect this wall of the stomach? Yes, but there is a mucus and bicarbonate ions. Mucus and bicarbonate ions. That means the mucus and bicarbonates of the gastric juice will protect the stomach, wall of the stomach. That means certain cells in the wall of the stomach, and also the wall of the stomach will secrete mucus that will lubricate the wall of the stomach, so that it can prevent from the acidic effect of the HCl. Here bicarbonate is a buffering agent of acidic substances. Here this is very important. The mucus and bicarbonate, they protect the mucosal epithelium from the excoriation effect of the acidic content, that means the HCl. Okay, then this is about the enzyme, and the same HCl will convert the pepsinogen into pepsin and the prorenin into renin. Right. But the thing is, in case of infants, there is a renin enzyme, because infants consume milk. Now it contains a protein called casein. The casein protein will be subjected to renin enzymatic activity and a calcium, that leads to calcium paracaseinate. And this calcium paracaseinate under the activity of pepsin, it gives rise to peptones. Here food is also formed. Okay, peptones. This is very important. Pepsin. Casein will be subjected by renin activity to form calcium paracaseinate, and the calcium paracaseinate will subject to pepsin to form peptones. And the most important thing is proteins. I told you already. In stomach, protein digestion will take place because of high concentrated pH 1.8, that means pepsin activity. The protein will be break down to proteoses plus peptones by pepsin. This is very, very important enzymatic activity. Proteins, that means the pepsin acts on protein and break down into proteoses and peptones. This is number one. Whereas renin act on casein, that means the milk protein, under the presence of calcium to form calcium paracaseinate. And this calcium paracaseinate subjected to pepsin activity to form peptones. So ultimately the peptones and proteases will be formed. The end product, that means the resultant enzymatic action, that means the enzymatic action leads to resultant products of proteins or proteoses and peptones in stomach. This is very important. What we get in stomach? In stomach, that, for example, in case of buccal cavity, we got bolus with 30% of carbohydrate enzymatic activity. That means breaking down. But here in stomach, we got peptones and proteases. Here there is no carbohydrate breakdown, there is no fat breakdown, there is no nucleic acid breakdown. Only the proteins are breaking down because of high pH content. Because the peptide bonds are so powerful that it required a strong, concentrated, highly concentrated medium. Because peptide bonds are the powerful bonds of biomolecules. Okay. So for pepsin, we require such a huge and highly concentrated pH. Okay, it's about digestion in stomach. Then, so we got it to peptones and proteins. The next part is digestion in small intestine. Digestion in small intestine. I told you already. Sequential digestion. sequential digestion, you know. Next, the third part, the most important part, digestion in small intestine. Here, just like the gastric secretions play an important enzymatic role in case of stomach, here in case of small intestine, the pancreatic juice and the intestinal juice. I told you already what is intestinal juice. Here the intestinal glands are two kinds, Brunner's gland and Cripps of Lieberkühn. See, intestinal glands, intestinal glands, because you know Brunner's glands and Cripps of Lieberkühn. Cripps of Lieberkühn. The secretion of the same leads to succus entericus. Succus entericus, what we call intestinal juice. Intestinal juice. Then the pancreatic juice. The pancreatic juice. This is very important. The pancreatic juice and the intestinal juice will facilitate the digestion. And the most important thing, before we go into the enzymatic role, we have to discuss about the conditions and requirements. You know, this the activity of, that means the enzymatic activity. The enzymatic activity of intestinal juice and pancreatic juice required alkaline pH. That means pH of 7.8. This is very important. For that, the bicarbonate ions, how they maintain? The bicarbonate ions, the glucose, they plays an important role. They maintain the alkaline pH because the enzymatic activity of, you know, the succus entericus and the pancreatic juice will take place in alkaline pH only. Just like the pepsin in acidic pH of 1.8, these two enzymes, that means from pancreatic juice and the succus entericus in alkaline pH 7.8. Say for example trypsin, say the chymotrypsin, carboxypeptidase, aminopeptidase, pancreatic lipase, what we call steopsin, the pancreatic amylase, for example, you know, there are different enzymes, but they perform, that means their catalytic role will take place in the alkaline medium. The bicarbonate ion plays in, that means secreted from the pancreatic juice. At the same time, you know, the bicarbonate ion, some of the bicarbonate from the duodenal cells, particularly in the proximal part, that means we will secrete so that it can neutralize any acidic content, say from gastric juice, will if it goes down. That means to neutralize the gastric juice, the further bicarbonate ions are also circulated by the, you know, the duodenal cells. So the objective of these bicarbonate ions and mucus, you know, is to, you know, lubricate and protect from the acidic effect. That means protecting the intestinal mucosa is very important. Protecting the intestinal mucosa from the acidic effect. At the same time, it also maintains pH of 7.8. The bicarbonates, they maintain pH so that it can facilitate the enzymatic activity of pancreatic juice and the intestinal juice, what we call succus entericus. Okay, these are about different conditions. And the pancreatic juice contain different enzymes, you know, like trypsin. I am just writing the active forms, okay? But initially they are inactive. Say trypsinogen, chymotrypsinogen, okay? Chymotrypsin, steopsin, what we call pancreatic lipase, steopsin, etc. That means pancreatic lipase is called steopsin. Just like salivary amylase as ptyalin, pancreatic lipase as steopsin. Okay, these are different enzymes of that means required in present in small intestine. We will discuss how digestion will take place in small intestine. This is very important. Say digestion in small intestine. We already discussed now. The chyme plus formed in stomach will goes to small intestine. And first here the trypsinogen, the inactive forms. The trypsinogen should become trypsin. How? Enterokinase will convert the trypsinogen to trypsin. And after that, some of the trypsinogen by autocatalysis form trypsin. What we call autocatalysis. You know, autocatalysis. Autocatalysis. Say the chymotrypsinogen. The chymotrypsinogen. Under the activity of trypsin, form chymotrypsin. We got it. That means the active forms, the trypsin, the chymotrypsin, both we got it, and they will act on and they will perform the next subsequent digestion. Okay. Say for example, say the proteins, the peptoses, proteoses, and peptones. Proteins, proteoses, and peptones. They are subjected to trypsin, chymotrypsin, carboxy, and aminopeptidases to form tripeptides. Plus dipeptides. I told you now. Sequential digestion. First the proteins, then the proteoses and peptones, which are the intermediate stages of proteolytic activity. Here the proteoses are complex. They are the complex molecules. Then these proteoses and peptones are further subjected to the trypsin. That means further proteolytic activity to form tripeptides and dipeptides. Tripeptide means here 3 amino acids and 2 peptide bonds. Dipeptide means 2 amino acids, 1 peptide bond. Okay, 2 amino acid and 1 peptide bond. So for the degradation, okay, then these tripeptides subjected to tripeptidases. Dipeptidases they release from the intestinal juice, that means succus entericus, to form dipeptides plus amino acids. Then the dipeptides subjected to dipeptidase to form ultimately amino acids. That's it. So the end product, we knew that amino acids are the fundamental pillars for proteins. We got amino acids, so the protein digestion is completed in small intestine. Then regarding other activities, carbohydrates. The last part now, so the rest of the things have to finish off. Carbohydrates. I told you already. 30% of carbohydrates breakdown takes place in buccal cavity by cell amylase or taurine. The rest will take place here. How? Say the first the carbohydrates. Say the carbohydrates will break down by under the activity of amylase. Okay, to disaccharides. Disaccharides means maltose, sucrose, lactose. Then after that we got it maltose. Say here maltose is a disaccharide supplied by maltase to form 2 glucose molecules. Then sucrase to form 1 glucose plus 1 fructose. Then lactose by lactase to form 1 glucose plus galactose. So maltase, sucrase or invertase and lactase, they convert disaccharides into monosaccharides. That's it. Carbohydrate is done. So we got amino acids in small intestine and we got monosaccharides, between the glucose, the galactose, the fructose, we got everything. And how about the fats? I told you already. Pancreatic lipase is called steopsin. But before that, there is another important thing called emulsification. Actually, bile juice doesn't contain any enzymes, but it is plays important role in emulsification. How? The glycocholates and thyrocholates of sodium and potassium, what we call bile salts, will play an important role in emulsification. What is emulsification of fat? It is a breakdown of larger fat molecules into smaller micelles, known as emulsification. And these emulsified fats will be subjected to, say, emulsified fats. Emulsified fats. How we get? Because of bile salts. These emulsified fats, this will be subjected by lipases. Ultimately, the fatty acids and glycerols are formed, but in a sequential pattern. Okay. First, diglycerides will be formed. Diglycerides. Then again the lipases to form monoglycerides. Then again lipases to form ultimately fatty acids plus glycerol. Done. We got it. Because with fat, that means the simple absorbable forms for fat is fatty acids plus glycerol. In case of proteins, the amino acids. In case of carbohydrates, the